The purpose of this lab was to discover the effect that catalase has on the rate of reaction. A catalyst is something that increases the rate of a reaction without being consumed by that reaction. It lowers the activation energy of the reaction. The catalyst in our experiment is catalase. Catalase is a common enzyme that catalyzes the decomposition of hydrogen peroxide into water and oxygen. The substrate in our experiment is hydrogen peroxide, which decomposes into the products water and oxygen. An inhibitor is a substance that reduces the activity of an enzyme, either by entering its active site or by indirectly changing its shape or efficiency. The inhibitor in our experiment is H2SO4, sulfuric acid, which will stop the activity of catalase. We hypothesized that the longer we allow the catalase to react with the hydrogen peroxide before adding the inhibitor, H2SO4, the more water and oxygen will be produced, and therefore less hydrogen peroxide will remain. This means that when we titrate the remaining mixture containing hydrogen peroxide and H2SO4 with the potassium permanganate, it will take a smaller amount until it is completely titrated.
This graph is a visual of our hypothesis, with the reaction time as the independent variable and the amount of H2O2 used as the dependent variable. Throughout the lab, we made sure to wear our lab goggles and latex gloves. At the start of the lab, we had our plastic cups labeled in time intervals from 10 to 360 seconds. Our first step was measuring out the amount of H2O2 that we would need for this experiment. We then used a syringe to place 10 milliliters of H2O2 into each of our cups. Then we moved on to the catalase. Using a pipette, we placed 1 milliliter of the cold catalase in each cup and gently swirled it. After the specific times for each cup had passed, we used a syringe to add 10 milliliters of sulfuric acid to each solution and gently swirled it in order to stop this reaction.
That's the control. Okay, so we can... I think so.
We then used a syringe to transfer 5 milliliters of our solution into a separate cup with the same time labels on them. After this, we began the titration by using a syringe to slowly add the potassium permanganate to each solution, keeping track of how much was being added. We continued to do this until the solution turned a purplish-pink color. This change in color indicated that no more potassium permanganate could be broken down by the H2O2 in the solution, meaning that there was none left in the solution. We used this titration process to measure the amount of remaining hydrogen peroxide left in the solution after the catalase broke some of it down. The amount of potassium permanganate it takes for each sample to be titrated will differ because each one reacted with the catalase for a different amount of time. We then organized this data and graphed it accordingly. As suggested by our very low R-squared value, the data we collected did not exhibit a strong correlation, which surprised us as they differ greatly from our hypothesis. There seems to be somewhat of a negative correlation, with the exception of the 60-second reaction, from the 10-second reaction to the 120-second reaction, which is what we expected. Between 90 and 120 seconds, the amount of hydrogen peroxide left remains the same, which corresponds with our understanding that reaction rate levels off as time progresses. Notice that beyond 120 seconds, the remaining amount of hydrogen peroxide actually increases from 0.3 milliliters to 0.5 milliliters. Perhaps because as time progressed beyond 120 seconds, the enzyme was denatured due to prolonged exposure to the room temperature environment, impacting the data we collected. Some sources of error in this lab could include the measurements we took for the substances, specifically the catalase, hydrogen peroxide, and potassium permanganate.